Welcome to the class. So we are into the next chapter, which is the sexual reproduction in plants. We all know that plants, higher plants, would undergo sexual reproduction. So in this particular chapter, which is the second chapter under the unit of reproduction, we have a topic on discussion of sexual reproduction in case of plants. Now, plants undergo sexual reproduction. Now, how can they undergo sexual reproduction without basically a sex organ, right? So, plants have flower as a sex organ. So, in this particular chapter, we are going to talk about the structure of a flower. Then we are going to talk about some of the basic mechanisms by which there are some events which we can call as the pre-fertilization events. We are going to discuss about that. Afterwards, we would also be discussing about the actual fertilization. Now in plants, I'm sure you would remember what we have learned in our last class, last year rather. We learned about double fertilization, so we are going to learn about double fertilization in detail. And after that, we are going to learn about the post-fertilization structures and events, right? Now, after all this, we will try to figure out how the sexual reproduction is slightly different. In case of plants, we talk about two important traits and characteristics that we see in plant, which is called as apomixis and polyembryony. So we will very soon learn what they actually are. Right. So talking about the first topic of how sexual reproduction happens in a plant, we would like to discuss about the basic structure of the plant flower, right? So we have already seen that in a plant we have a structure. Is the higher plants we have a structure which we call as the flower, and the flower will generally have the region where some kind of structures like this, generally which are green in color. Then we will have the bright colored petals, right? So this would be our sepals and this would be our petals. So there are two walls that we have written, the outer wall and the inner wall, right? Now below, just inside this inner wall is what you see, a very important region of the plant or the sex organ of the plant, which we call as the andrecium and the gynecium, right? So here we would have a structure similar to this. So we would have a structure very much similar to this. Which is the female reproductive part, which we call as the style, stigma and the ovary, the style, stigma and the ovary, which is the gynecium, right? So here we have a region which we call as the ovary, then we have a covering to that which we call as the ovule, and then we have a region which we call as the stigma, and then we have a region which we call as the style. So all this is our female reproductive system. Now along with the female reproductive system, we have the male reproductive system here, which would look like these are all actually filaments, so they are all bilayered, but I am going to write one single tube like structure, which is the filament. and on that we would see the pollen grain, right? So this is the androecium, which has the pollen grain that is present inside. So we have seen that a flower is actually made up of the male reproductive system and the female reproductive system, both of them which are present inside this flower, right? So this basically is the sex organ of a plant, right? And now in this particular class, what we are going to find out is to see how each one of them gets, kind of this structure, and how this actually helps to form the seed which helps to produce the offspring later, right? So let us just go on to find out how this happens in the same way as we learnt in our last class. So in our last class we divided the whole of this into different stages, right? So even now we are going to different stages of the plant. So we will start off with the first one, which is the pre-fertilization events. So we know what fertilization is. We have already seen that fertilization happens when the two gametes would fuse, and we have seen the structure that there is androecium and the gynoecium. They both come from the flower, and there is transfer of this male gamete into the female gamete, right? So this is what we have already seen. Now, even before we actually see the flower on the plant, the plant is actually decided which part of the plant, which part of the cell is going to be a flower later. Okay, so that what we spoke as cell differentiation, right? Now that has already started to take place. There is already differentiation, and there is a particular structure where the flower would develop. Now this region is what we call as the floral primordium. Floral primordium basically is that group of cells which have undergone cell differentiation, where the cells have decided that they should bear the flowers. But not always we do see flowers, right? Many times we have also seen that there can be inflorescence. So there can be inflorescence, or there can be flowers that would develop from this floral primordium. Now this floral primordium would undergo differentiation, and this would give us a flower, right? Now let us do one thing. In this floral primordium, let us see how the structure of the androecium looks like. Okay, the floral primordium is there, from there comes the flower. So now what we would do is we would talk about the post fertilization events, where we have understanding the structure of the stamen. Then we will also understand the structure of microsporangium, right? And then we will also discuss about understanding the structure of the pollen grain in today's class. Okay, now basically a stamen is having two important parts. Okay, so if I am talking about stamen, stamen is the most important male reproductive part of the flower. So we have a stamen, and this stamen is kind of divided into two important regions. Okay, so the first one is called as the filament, and the second one is called as an anther. So let me see and show you how this actually looks like. Now filament, as the name itself suggests, it's a very long and slender tube, Okay, so it's basically a long and slender tube which would be the whorl of the flower, okay, which would be the whorl of the flower, and then you have the anther, which is a bilobed structure. Okay, this is a bilobed structure. So there is a bilobed structure of the anther. So anther is always two lobed like this. It's the bilobe. Now always you see that the proximal end of this stamen is attached to the thalamus, this basal part, it is attached to the thalamus or it is attached to the petal of the flower. I am sure if you have, when we have done the dissection of the hibiscus in our last year, you would have seen that when you take out the anther, you are you see that it is attached to the petal of the flower, right? And this is what makes the whole of the stamen to be inside the flower in a firm place. Okay, now comes the most interesting part. The number and the length of the stamens are completely different in different species. In fact, you can count the number of stamens in each flower, and you can kind of tell which species it would belong to. At least you can make a wild guess there, right? So there is high variation in the entire presence of anthers, and if by chance we have a very good microscope, we can actually take these anthers, try to see its structure, and we try to understand how it looks like. Okay, now that is something which is very, very important. Okay, now most of the times you see that there is a bilobed anther. Now bilobed anther is a characteristic of angiosperm. Okay, we call that as theca, we call it as dithecus. If you remember what we learned in our last class, we call this as dithecus. Now you see that in this there is one longitudinal groove that exists. Okay, there is one longitudinal groove that exists in that anther, which divides the anther into two different regions. Okay, now if by chance, I would like to take an anther and kind of take a section of that, a transverse section of it. Suppose this is the one, I would take a transverse section of this anther. Then I would see a four-sided tetragonal structure. Okay, then I would see a four-sided tetragonal structure of anther. Okay, tetragonal structure of the anther. And if I just look at that, you would see how beautiful that would look like. Okay, now if I just write a rough diagram, it would look very much like this, very much like a butterfly. Now you can see that this kind of goes inside. Okay, now depending on the development and all, this groove can go slightly longer. Okay, so this is how it would look like. Now this will have many layers. This would be one layer, and below this there would be one more layer. Okay, this would be one more layer. So there are two layers inside, and you see that here there would be some kind of circular structures that are present. Okay, some kind of structural force structures which are present. Then this is where a very, very important important process of production of spores takes place, production of anthers takes place. Okay, so let us see what these structures are actually called as. Okay, so this outermost layer is what we call as epidermis, right? Then just below this we have the second layer, which we call as endothecium. We call that as the endothecium. Okay. Now there is these kind of structures, a structure which is circular here, right? Now this circular structure has some mass inside, some kind of cells which are actually involved in the formation of spores. Hence we call this as the sporogenous tissue. What do we call this as? We call this as the sporogenous tissue, and in the sporogenous tissue is protected by another layer which we call as the tapetum. So basically the whole of the anther, if I take a look like this, it would look somewhat having this kind of structure. Okay. So what would happen in this sporogenous tissue? Now in the case of sporogenous tissue, we see that there is formation of spores. Okay, these are called as spores, means they produce spores. Now these spores which are formed are very small. Okay, and hence we call this as the microsporangia. We call this as micro. Why do we call this as micro? Because we are talking about the male reproductive system, and we know male reproductive system is very small, right? So we call it as microsporangia. Okay, so there are two in each lobe. So this is half lobe, this is half lobe. So you have two microsporangia in the lobes, which is generally at the corners. Okay, now this microsporangia is very important, because from microsporangia what we get is the pollen sac. Okay, we have a region which we call as the pollen sac. Now pollen sac is like throughout the anther. Okay, from top to bottom you see this pollen sac that is present, and these pollen sacs have the pollen grains. Okay, these have the pollen grains. Okay, now let us see what is the structure of this microsporangia. This microsporangia has the epidermis, right? It has the epidermis, then it has a endothecium. In that there is the sporogenous tissue, and here there is formation of the pollen sac, and then there is formation of pollen present inside. Okay, now let us see how this microsporogenesis happens. Okay, I am going to erase this part of our basic structure now, and I would like to tell you about how microsporogenesis takes place. You know, microsporogenesis starts from this tissue, the sporogenous tissue that is present in this microsporangium. Okay, so there is the cells present here. Okay, now these cells will undergo the first kind of division. The cells undergo division. Please remember that there is a tapetal layer outside, and this tapetal layer is kind of protecting the tissue, right? And the cells which are here are kind of undergoing lot of changes, lot of movements, and these cells which are undergoing cell division are not mitosis, but they are all undergoing the meiosis division, right? They are all undergoing the meiosis division. Now we know what happens when a meiosis occurs. You know that there is one parent cell which would give rise to four daughter cells, four offsprings, four daughter cells, I would say, not offsprings. So we have the four daughter cells. But now there is a, kind of, I would say, a catch. Now instead of forming four different cells, what you see is this division leads to one cell which we call as the microspore. Okay, it leads to a microspore, and microspore is always a tetrad. Okay, microspore is giving rise to a cell which we call as a tetrad. You know what is a tetrad? Tetrad is 4 different cells. So there basically it's something like, you know, the cells which are here would look like 4 different cells which are fused together. Okay, so basically this is what we call as the microspores, where the haploid cells are as tetrads. Okay, we call this as a microspore tetrad. Please do remember that they are all present in our anther now. Now the anthers will mature. Yes, as the flower matures, you see that the anthers also mature, and when the anthers are mature, there is one step of dehydration in the pollen that would happen. So basically it's going to dry up. Okay, so they won't be fluffy, and you see that this matured anthers only would undergo this process of dehydration, and at this point of time the microspore tetrads, you know, they are going to get dissociated from each other, right? They are going to get dissociated from each other, and when they get dissociated, they, after, I would write it here as a process of dehydration, after a process of maturation. Okay, after a process of maturation and dehydration, they would you know, kind of separate from each other, and then they would form the pollen grains. Okay, then they would form the pollen grains. So if I'm just talking about one particular anther, one particular microsporangium, one microsporangium has 4 sporogenous tissue, and each sporogenous tissue will have thousands of microspores, and those thousands of microspores would finally give me thousands of pollen grains later, right? So this way, from the stamen we have the anther which has the sporogenous tissue protected by the tapetal layer. This would undergo maturation, dehydration, desiccation and all that, giving us the pollen grains. Now from the pollen grain there, we are going to see that the male reproductive system is formed. Okay, so let us start our understanding of how this pollen grains are. So talking about pollen grain, you know, these are nothing but our male gametes. Pollen grains are our male gametes. Okay, not gametes, actually gametophytes. Okay, pollen grains would give rise to uh, uh, pollen. Pollen will have the male gamete, right? So instead of writing it as gamete, let me put it as gametophyte itself. You will understand and remember it better that way. Okay. Now this pollen grain are already coming from a dried part of the flower, right? It's already dehydrated and desiccated, right? So when it is already dehydrated, when it is already desiccated, you see that they can come out of the anther very easily. Okay, you can see that they would come out very easily, and if by chance you try to take this kind of pollen grain, put it on a slide and add slightly some kind of sugar solution on that. Okay, you add some sugar solution, you would be astonished to see how beautiful the pollen grains are. And not only that, you can also see that the pollen grain changing its structure. Okay, because you see that they are actually initially round, they are very, very small spheres, say something like, you know, around 25 micrometers, right? You just see the presence, it has some kind of yellow dot if you at all you touch in your hand. Okay, but if you see it under the microscope, you see very clearly that they have different structure. Okay, now generally you see that this pollen grains have two membranes. So one is outside and obviously the other one should be inside. Okay. So we have the outer layer which is called as sporopollenin. Okay, the first one which is called as the sporopollenin. Okay. So it would be like, I'll just write this as a very soft spherical structure, but actually it is not soft. You see that it is very rough, has some kind of spikes and spines on its body. Okay, so I would just put it like this. Okay, just for our reference. Okay, now this is made up of one chemical which we call as the sporopollenin. Okay, this is the outermost membrane which we call as exine. Okay, the outermost membrane is called as exine, made up of sporopollenin. You know what? This is the organic material and considered to be one of the most hard, resistant, heat resistant, temperature resistant organic material that is known to us. Okay, you can put it in water, you can put it in high temperatures, you can put it in acids, you can put it in alkali, you can put it in most of the enzymes. You see that it does not get damaged at all. Okay. However, you see that there are small pores, okay, small regions present, you know, on this exine where this sporopollenin is absent. Okay, say for example, let me say I'll just write it here. So there is one small region where sporopollenin is absent, and those kind of very small tiny pores is what we call as the germ pore. Okay, we call them as the germ pore. Now here sporopollenin is absent. Now you see that this is a very important region. I am sure you will be understanding it very soon now when we go to the next step. It is at this region where there is lot of structural changes that would happen inside a pollen grain. So there is exine, there is the presence of the germ pores on the exine. Okay. Now you know, there are so many different types of structures that are present here. Then inside this exine, you know, you have an inner membrane, and this inner membrane is what we call as intine. So outside is exine, the inner one is intine. Now intine is not very thick as exine. In fact, it is actually very thin. Okay, it's a very thin membrane. It's not as rough as exine. It is very smooth membrane also. Okay, and inside this you see that there is cytoplasm. Okay, if outer exine is made up of sporopollenin, inner one is like our all the other plant cells, which is made up of cellulose, which is made up of pectin. Okay, so let us see what is there inside this. You see the presence of cytoplasm. Okay, the cytoplasm is there, and then there is a plasma membrane. Now suppose we have a cell like this. Okay, we have a cell like this. You see that the pollen grain was a tetrad, right? It was a tetrad. So this tetrad would undergo cell division, right? So there is a plasma membrane, there is cytoplasm. Now when the pollen grain is mature, you see that it does not look just like one single cell. If you look at, kind of take a cross section, you would see that there are two cells that are present inside. Yes, there are two cells. So we have one cell here outside and one cell which is in the other side. Okay, now this cell which is generally you know, at the base, can't say it's a base because we are talking about a sphere, but something which is slightly heavier, much smaller also, has a much smaller nucleus, we call such a cell as the generative cell. Okay, we call such a cell as the generative cell, and the bigger one is the vegetative cell. Vegetative cell, and vegetative cell also has a nucleus. Most of the times we know what vegetative means. Vegetative means something like of the body, right? So it is always bigger. It has lots and lots of food which helps in the uh, growth of this generative cell. Okay. And has some kind of nucleus which is irregularly shaped. Now generative cell is very small, kind of dense, okay, and keeps moving inside the cytoplasm, okay. So you see that this is how there are two cells inside our pollen grain, okay. Now in most of the angiosperms, what would happen is at some point of time when they are mature, okay, you would see that the two cells would undergo mitosis to give us two male gametes, okay. So there is this vegetative cell, right? So there is this vegetative cell. You see that they would undergo division basically to see that in this whole of the pollen grain you would have three different cells that are present. We call that as the three-celled stage, okay. We call that as the three-celled stage. Now pollen grains are a very interesting characteristic. I'll tell you why they are interesting. Because first thing is that they are very small, but they have the ability to give rise to work as a male gamete. But you remember that I did not write it as gamete, I wrote it as gametophyte because the entire pollen is not a gamete. In this entire pollen, okay, basically there are two cells, inner and vegetative and the generative. Now in the vegetative again you see that they would undergo cell division, there would be two cells that are formed, generative there, and of course that is depending on species and many others, but you see that there are two cell stages and three cell stages. But whatever said and done, it's not that both the vegetative cell and the generative cell is going to give rise to a plant. They are not working like a male gamete. So only a part of our pollen grain is working as a male gamete, and hence I would not write it as a male gamete, but I would write it as a gametophyte. Okay. Now in our maybe next class, I would tell you about the female reproductive system of the flower, and then we will talk about the fertilization. In fact, this three-celled stage is something which is very important for discussing about the syngamy or the fertilization that happens inside the flower. Okay. Now since we are talking about the pollen grain, I would like to mention to you about some of the problems that these pollen grains would cause. When it is completely dehydrated and desiccated, this pollen grain would come out of the flower by a slight wind or by air, and it will be in our atmosphere. I told you that they are also lightweight. So what would happen is they would kind of keep moving in the air, and by chance we breathe them. It might cause lots of allergies, lots of bronchi problems, asthma, okay, chronic respiratory disorders, and so on. Best part was the Parthenium which we spoke of, which causes pollen allergy, right? But however, please remember that this vegetative cell has lots and lots of nutrition. Now because it has lots and lots of nutrition, you know what, people would actually take all this pollen grain and use it as food supplements. They say that it is very important energy booster for athletes, though I don't know if it is legalized and it is safe. However, that is for us to do a lot of research later. Okay. So we have also seen that these pollen grains, once they come out, they would fall on the stigma, then there would be some kind of development that would happen. But now I have one question that I would like to ask you. Do you think that this pollen grains would stay in, if it comes out of the flower, do you think that they can sustain their viability, say for months, years, or whatever? What do you think is the lifespan of this mature pollen grain? Now that is my question to you. So you can kind of think about it and answer to me about this in our live class. See you later. I hope you have enjoyed the class. In our next class, we will talk about the female reproductive system in a flower and discuss more about the pre-fertilization events in the case of plants. See you till then. Bye.